Hi everybody, I'm Jason Fernesiak. I'm one of the Interactive Associates for Fertility and Sterility, and I'm here with Dr. Emre Seli. Dr. Seli is a professor of reproductive endocrinology and infertility at Yale University, and is also the Chief Scientific Officer for EVRMA Global. We're here to discuss his prize paper abstract, the developmental potential of human aneuploid embryos beyond implantation. Now, Dr. Seli, I understand that this manuscript just recently, moments ago in fact, was awarded the prize paper for the ASRM 2019 Congress. Congratulations.
Thank you. We're very happy.
I can imagine so. Would you be able to tell me a little bit about the abstract, what the findings were?
Yes. Well, we're really delighted to have been recognized for this work, and we are very proud of this work because it required a lot of different investigators to collaborate in a quite selfless manner and across continents. I would like to emphasize that two key investigators in this project were from Cambridge, Marta Shahbazi, and also Magdalena Zernitschka-Goetz. And also other very key investigators are Richard Scott from Army, New Jersey, Tony Pellicer from Rome, and a number of lead investigators, also Xin Tao and Tianren Wang. We were able to bring a group of people with different expertise to ask a question and to answer a question that we could not answer alone. And I think this is one of those examples of studies that we need to do collaboratively in order to answer important aspects of reproductive medicine and practice that again cannot be done alone and cannot be done with one type of funding. We wanted to gain insight into the differential development of euploid and aneuploid embryos into the second week of existence. And at this point we saw differences in the development of trisomy 16 and monozoid 21 embryos, and then we were able to identify a potential mechanism by which the trophoblast development is suppressed in trisomy 16 embryos.
So I understand that there was a fairly novel technique that was developed for culturing embryos into or past the implantation phase. Can you tell me a little bit about that?
Yes, this method has been developed and published in Nature Cell Biology by Marta Shahbazi and Magdalena Zernitschka-Goetz, and allows us to look at development of key tissues such as epiblast, hypoblast, which will then lead to certain tissues that are of interest. Therefore, it allows us to investigate specific deficiencies in these tissues, specification at different stages of early development.
What are the implications of this study for reproductive biology?
Well, I think here we wanted to do things that will kind of, remain after we're gone or retired, etc., and maybe form the basis of people in understand the role of extra or missing chromosomes in the set of 46. That is I think, importance of this specific study. I guess we established a model on how it can be done. Other people can repeat this in order to study different aneuploidies and understand why they lead to loss of pregnancy or congenital abnormalities. In the future, we are hoping to expand our understanding of specific aneuploidies, but more importantly, we want to expand our research to the study of mosaicism. Because I really think, although we discuss mosaicism a lot, the implication of mosaicism on pre-implantation testing a lot, but we don't know much about mosaicism. What happens to aneuploid cells in a mosaic embryo, or do aneuploid cells in a mosaic embryo divide or don't divide? Where do they go? Coming from a laboratory who uses animal models, I don't believe there is a really reliable animal model to study human aneuploid and human mosaics. So we have to do this in human embryos.
Really very fascinating work, Dr. Seli. Congratulations.